We said before that how your neurons, your different cells in your brain, came out. So we said that after 3 weeks of conception, you have the embryo, which is divided into ectoderm, mesoderm, and endoderm. And there are inducing factors which become nervous tissue. So you see it starts, your nervous system starts with a blastula. Then it has several, processes. Then it becomes, the neuron becomes a neural stem. Then it becomes stem cells, neuroectoderm, stem cells, then it becomes a neuron. So it goes to a process, in order that your nervous system become completely formed. So it starts with an ectoderm, neuroectoderm, stem cells, and become a neuron. Ready. So these cells migrate, laterally and become also melanocytes. So you can see that melanocytes come from your cells from your nervous system. And it also become the dorsal root sensory ganglia and sympathetic chains. So you can see that from neuroectoderm, again, it becomes stem cells, then it becomes your neuron. And this neuron, the cells migrated to become melanocytes or become a sensory ganglia and sympathetic, peripheral nervous system, as well as the ganglia of the enteric nervous system. So it also gives rise to tissues, including your neuroendocrine, cardiac, mesenchymal, skeletal systems, forming the basis of the many congenital syndromes involving brain and other organs. Meaning to say, even if they are still, the human being, that's why your human development inside your stomach, what you call that? The baby inside the womb, you can observe that it is very, very sensitive to any changes because it is at this portion that different organ systems are being formed. So that's why it's very dangerous at this stage. You can see that from very micro cells, there are many processes and it goes to many forms of your nervous system. Then you have another non-neuronal structure, and which you call that your notochord. Notochord plays a critical role in your neural tube differentiation. Then, when the neural tube is formed, it develops nervous system cells into a rapid number. So it is at this time that when neural tube is formed, there is a great number of nervous system cells that is being formed. Then it goes to the other layers of your nervous system. So you can see that there is proliferation of your neurons to other centers and become, it goes to different dimension. The longitudinal dimension, the circumferential dimension, and the radial. This is already the neural tube. The longitudinal dimension become your brain and spinal cord. The circumferential dimension become the dorsoventral axis and medial to lateral axis. Finally, the radial dimension becomes your innermost layer adjacent to the ventricles and outermost surfaces. So you can see that there are 3 dimensions, which gives out the different parts of your nervous system. The longitudinal is your brain and spinal cord. The circumferential is your dorsoventral axis. Then the medial to lateral axis, and the last is your ventricles and to the outermost surface. I will not go deeper on that since I will just give you a copy. We'll just discuss the more important one. In spinal cord, the majority of tissue comprises lateral plates. So your spinal cord comes from your longitudinal dimension. It divides into dorsal or alar plates, which compose of your sensory interneurons and motor or basal plates consisting of your ventral motor neurons. Diba yung dorsal, ventral, motor neurons. Your floor plate, in response, it is located in the notochord and produces to become the factors that specify cells' phenotype and function. So your notochord is the one that gives rise or expresses the phenotypes or phenotypic function or phenotype portion of the human being, the genotypes and phenotypes. Your phenotypes is the one that expresses the human being, the physical expression, and the genotype are your DNA and RNA. So it is from the notochord. Your phenotypes are from notochord. So let's go to your migration of your nerves. There are 3 well-recognized spatiotemporal patterns of neurogenesis that underlie regional brain formation. You have 2 radial patterns. However, you have the third, the non-radial or tangential migration of cells. So your cells, meaning to say, migrates into different portion, into spatiotemporal pattern, into radial pattern, and into non-radial or tangential pattern, which later give rise to the different zones of your brain. So you see that at the end of this processes, you can see that there is also the destination of your different nerves. So it travels along the processes of your radial glial cells. So meaning to say your neurons has migrated to the different portion of your brain, and it has a final destination, meaning to say your brain now becomes or have a different portion or parts as your neurons develop and migrates. So, however, you have also what you call cell death. No, aside from being, we said that there is proliferation, there is what you call that, giving rise to the different portion of your nervous system. However, there are also cell death. No, so I will not go to the very specific on the different cell death, but it is important to know that there is also cell death as they proliferate. So here you have how your genes or the expression of your genes are also being taken care of in your development of your neurons. It also contributes to your normal development and brain function. Your RNA regulates the RNA transcription, alternative splicing, molecular modification, and RNA translation. So there are a portion of your neuron that are part of proliferation. It becomes, there are cell death, and there are some of expression of your neurons that would contribute to the expression of your neurons, your normal development and brain function. So, as I've said, your cells also migrate. No? Migration of cells into the neural crest, and then it goes to your new location and the different portion of your brain or in the different portion of your nervous system. No? So they have the final destination as they migrated. So let's go to synaptic genesis, no? So you have, there are processes, as I've said. There is what you call aggregation, there is migration, no? There is proliferation. So here, it is what you call synaptogenesis, no? There's an aggregation, complete aggregation, axons and dendrites grow out from the neurons. So that is what you call synaptogenesis, no? So when the neurons are already formed, after proliferation, after migration, there is already what you call that aggregation or spread of your axons and dendrites out from the neurons. Axon growth is directed by growth cone at the growing axon tip, meaning to say it divides, no? Your neurons divided into many axons and dendrites. So there are different ways in how your neurons divides. So let's go to how your different parts of your brain are affected during the neurodevelopmental of your different neurodevelopmental basis of psychiatric diseases. So you have your neuron starts from the stem cells, then it becomes your ectoderm, stem cells, and becomes your neuron. So during this time, you have your proliferation, you have your migration, and you have the final destination. However, on the way, on the proliferation and the final destination, this is the time wherein diseases are developed, no? So you can observe that in schizophrenia, no, there is a smaller prefrontal cortex and hippocampus and enlarged ventricles. So we did not go in depth regarding how your prefrontal cortex was formed, nor your hippocampus. You already know that in your embryology, how your prefrontal cortex was being formed, how your hippocampus was being formed, and your ventricles. However, these are the part of your brains that plays part of your schizophrenia. Maybe you will not appreciate now what is schizophrenia. When a person has schizophrenia, the patient has hallucinations, delusions, and totally the patient cannot work or cannot do complex thinking. So at the end, they cannot really work. There are some, but in complicated cases, generally schizophrenic people cannot work. That is because of your different parts of your brain, again, which is your prefrontal cortex, your hippocampus, and enlarged ventricles. Then your other psychiatric disease is your autistic, or your autism spectrum disorder, So there is less or reduced of gray matter, disorganized laminar patterns, misoriented pyramidal neurons, and ectopic neurons. So you can observe that nerves are very important or have a very major role in the development of your different psychiatric diseases. So it is said that during the proliferation or during the formation of your nervous system, there should be a balance between the formation as well as the death of the neuron. Otherwise, there would be also an abnormality. So you can observe that there is balance between neurogenesis, death of unwanted cells, and adapted synaptogenesis. So proliferation, migration, death should balance or be equated. Otherwise, there will be an abnormality. So you can see that this is a portion wherein the brain is not fully developed. So you can see the persons or the human being whose brain are not being fully developed, especially the cerebral hemisphere. So I will not go deeper in that. So you have different abnormalities of the different portion of your brain when it is not being formed. Like your neural tube, it causes spina bifida or anencephaly, Then you have genetic disorders. Then you have environmental toxins. So these are different factors that affects the formation of your brain or the development of psychiatric illnesses, It's not only the migration, the proliferation, but also there are toxins or abnormalities in the formation of your brain. So it affects really the nervous system of a person even inside the womb of the human being. It doesn't mean that schizophrenia will only be developed or other psychiatric diseases can only be developed when you are outside the womb of your parents. Meaning to say, psychiatric diseases can be developed even inside the womb, even when you are still inside the womb of your mother or any human beings that is being developed inside the womb. So it is multifactorial. So that is spina bifida. So let's go to the neurophysiology.